Good day! In this video we will be taking a long hard look in the mirror, at this glass jar, which has been closed airtight for 4 years now. As you can see, it is filled with plants and a bunch of slimy green growth. But how did we get here? Well, we'll have to go back in time to a little over 4 years ago, when I managed to catch about a dozen fruit flies using glass jars stored in the freezer. They would need a nice house, of course, so I took this much bigger glass jar, added 2 nice plants, Coffea arabica, which I knew from experience would probably do well in a closed and humid environment, and Iresine herbstii, which I had no experience with whatsoever, placed in a little pot right in the center of the jar. As a source of food, I added a banana peel as well as a banana beer yeast smoothie, all of which fruit flies really like to eat. And in they went, the Drosophila melanogaster. Quite a pretty home, if I do say so myself. A day later they were flying around in their humid home, and another day later the smoothie was starting to form a moat around the plant pot, and starting getting increasingly moldy over the next days. After a week, the plants weren't doing great, but the fruit flies were, because they had already procreated, with loads of larvae crawling around. After only a single week, the first new adult fruit fly hatched, the first to be born in the closed ecosystem, and the amount of pupae boomed. More were definitely on their way, as can be seen by all the tiny larvae on the banana peel. 2 days after this, the plants, but especially the Iresine herbstii, also known as bloodleaf, were in really bad shape, but that didn't bother the fruit flies. 11 days after this closed ecosystem was made, this airtight jar was filled with flying insects. Just 2.5 weeks later, the jar looked completely different and not particularly pretty anymore. The bloodleaf almost completely died, with just a few tiny living leaves growing. The coffee plant looked a little better, but also had a lot of dead leaves. The glass was filled with dead flies sticking to it. Some of these are the first flies that were added in here, the pioneers if you will, and those died of old age. However, many of them are flies that were born here, about 3 weeks prior, and although they could have died from old age, their lifespan spanning from usually 4 to 6 weeks, more was probably at play. I think I added way too much dead organic matter in the form of the banana peel and smoothie. As we saw, it quickly started decaying. This process might have released toxic gases and potentially even depleted the oxygen levels. Not to an extent that everything was killed, as the plants are still sort of alive and there's also still some adult flies as well as larvae, but it could have killed a significant part of the population. I'm still not sure what really happened. At this point, the green transformation of the jar was starting. A lot of algae started growing on the glass, fulfilling the same role as the plants, producing oxygen while taking up carbon dioxide and turning inorganic nutrients into organic molecules, aka food. This was nice to see, as our plants really weren't doing well. 3 months later, the coffee plant seemed to be doing a little better, with many new leaves growing, and miraculously, the bloodleaf is still sort of alive. Moreover, an entirely new plant started to grow. The earth in the plant pot was covered by moss, which must have emerged from some spores or tiny plant parts in the soil. And, unfortunately, at this point all the fruit flies had died, for whatever reason, thus marking the end of the fruit fly presence in the fruit fly jar. Possible cause of death? Banana overload. This did not mark the end of the ecosystem itself, however. In fact, it was still developing. All of the moisture that was inside the banana and the smoothie, as well as in the soil of the plant pot and to some extent in the air, has now formed a surprisingly deep layer of water, especially after all the fruit fly food decomposed. As you can see, it is not exactly sterile. It is filled with these tiny white organisms, while actually they are really large for what they are at least. These are rotifers, a large group of microbes, many of which aren't visible with the naked eye. They probably emerged here as some, or at least one individual managed to make its way into the water, likely from the soil in the plant pot, as a resting egg, which can actually stay dormant for several decades. The water provided ideal living conditions, with plenty of food, without any competition. This led to explosive population growth, all stemming from one or just a few individuals. Pretty cool. But there's more. There's been another population explosion of a completely different species. These tiny dark globular springtails, which are very cute. They probably have a very similar story to the rotifers, just not an aquatic story. And these are actual animals. Like the rotifers though, springtails can reproduce through parthenogenesis, or asexual reproduction, so basically cloning. Like the rotifer population, this entire springtail population could be the progeny of one individual, or just a few. And these two saw a lot of food and little competition. And these two probably came from the plant pot soil. Whatever killed the fruit flies did not kill the springtails. Or maybe the springtails are here now because the conditions in the jar are livable again, but there's just no fruit flies left to do the living. Another 2 months later, when the ecosystem was half a year old, not much changed, except for the emergence of an entirely new plant species. And another 4 months after this, for whatever reason, both the plants were doing really great, with lots of new growth. The tiny new plant had also grown and the moss also looked very healthy, maybe because it was spring. When the ecosystem was a year old, both plants were doing great. In fact, the bloodleaf had now grown bigger than the coffee plant and even formed an aerial root. The new plant was also still there, but the moss wasn't looking great and had partly turned brown. The layer of water was still filled with algae, but not completely filled with rotifers anymore. They didn't go extinct, but I think their population crashed, because it grew too quickly in the first place. They could reproduce really quickly because they had no competitors, until they became their own competition. Interestingly, the same did not happen to the springtails. There's still a bunch of them left. And even after a year, a new species managed to appear. These are mites. There were a few of them in the jar, but not a whole lot. These just might be predatory mites, in which case they just might prey on springtails, which would be a really interesting dynamic. Wow, quite a history, but this was all information you might have had already. So, let's go back to the present and look at this ecosystem 3 years after the latest documented events. I don't have to tell you that a lot can happen in 3 years, even in these closed environments which, perhaps luckily for them, form a world of their own. Not interacting with the ecosystem you and I live in, but existing as little ecological wonders encased in transparent rock. And I feel honored to host some of them on my windowsill. Anyway, let's not get too sentimental too quickly and look at the jar. By now it's looking a little more wild. This is the side of the jar that wasn't facing the window for the past, I don't know exactly how long, but for the past period of time. As you can see, there's now a large network of aerial roots growing from the plants onto the glass and also down into the layer of water. Years ago, we saw the first aerial root coming from the bloodleaf. As far as I can tell, all these roots come from that same plant. The reason it's growing so many aerial roots probably has to do with the fact that the soil in the pot is very moist. If it's too wet, gas exchange can be made difficult, so it resorts to growing aerial roots. Nifty. This side of the jar is also covered in some sort of slimy growth, at least partially filled with algae, likely made up from the remnants of the dead flies and leaves that were on the glass. It would be really interesting to take a sample of this to see what microbes might be living in the slime, but I don't want to open the ecosystem, so you'll have to resort to your imagination. This is the side of the jar that was facing the window. There's almost none of that slimy substance here, and also way less aerial roots. This is probably because this part of the glass has been warmer most of the time, especially in summer. More water evaporated here, and it was a lot drier. Also a result of this side facing the window is more light, which resulted in a lot more plant growth on this side, but more on that later. The moss that was growing on top of the soil in the plant pot seems to have almost completely died, but the moss itself didn't. It's actually now growing in the little dish under the plant pot. There's still some living moss hanging from the edge of the pot, but it's mostly growing here, accessing the nutrients from the soil in the pot. There's even some moss growing out of the terracotta dish itself, using it as a substrate, which is pretty cool. It's a bit difficult to see unfortunately, because the glass is quite dirty, but I hope you can somewhat make it out. The dish is now a fuzzy green living thing. From the way the moss is growing, you can see the jar had the same orientation for quite a while, growing almost horizontally towards the window in some places. Behind the plant pot, which is again really difficult to see, there's even more moss growing, probably because it's darker and wetter, which mosses like. And again, for some reason, it stopped growing here. Now for the plants. Here we have the coffee plant. It's doing just fine. There's a lot of dead material, but also a lot of healthy green leaves. The conditions in this closed ecosystem are far from ideal for this plant, but good enough. And here we have the Iresine herbstii, the bloodleaf, which is actually kinda doing great, having grown towards the glass. All the leaves you see are from the bloodleaf. It also grew towards the top of the jar here, and we can see something interesting. Every leaf here is from the bloodleaf, even the really small leaves, which there are a lot of. Normally this plant doesn't produce such leaves, and certainly not this many. They're looking quite different from normal leaves of this plant. They're also much greener than usual. As the name suggests, this plant usually grows red leaves. Though I must say this individual already had quite green leaves when I added it to the jar. Some of the lowest leaves still kinda look like the original leaves. It's really cool to see that the plant is totally adapting to its environment, both with the shape of the leaves as well as all the aerial roots. Here you can see the coffee plant a bit better and the red stems of the bloodleaf. Both these plants almost completely died in the past, and yet they're still here. This part of the glass is almost acting like a plant as well. The slime is so thick that it is able to house enough algae to form a dark green blob, spanning almost the entire height of the jar, maybe supported by the roots on the glass, making it look like one solid organism, even though it isn't. Here it is a bit closer. This algae streak is fulfilling a very similar role to the plants. If those, for whatever reason, do die someday, these algae probably won't, and just might be able to support the ecosystem on its own. This jar isn't very aesthetically pleasing, nor will it win any beauty prizes, but it sure is biologically active, which is way more pleasing in my book. While filming, I suddenly noticed a lot of these tiny black stringy structures. I thought I'd never seen them before, but rewatching the old footage, I did notice that they were here before. There's much more of them now though. Now I hear you ask, what are these? Well, how should I know? Wow, relax guys, it's just a video about some dirty old jar. No, just kidding. I'm curious as well, because I do not know. They can be seen throughout the entire jar. My best and most of all only guess is that these are strings of springtail poop. But that would mean the springtails produced huge logs, because look at how small these cuties are. Oh, that's right, the springtails are totally still here, doing little springtail dances. Aren't they just the best? Just look at them go! I guess one difference between the algae slime and actual plants is that the springtails can feed on the algae much easier, which is a good thing I guess. And the jar is completely filled with these tiny six-legged animals, which is great news for all of us springtail lovers, oh yeah! These two seem to be on a special mission. Just look at that speed! Very impressive. Where are you going, little buddy? Got somewhere else to be? Ha 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 ha. There seems to be some sort of biofilm growing on the glass here, which this springtail seems to be grazing on. Those yellowish greenish balls on the glass might also be springtail poops, or something else again. There's so much we don't know. How mysterious. Another mystery is the species of springtail we have. Identifying them is made difficult by the fact that we can't see them from very close, the glass is very dirty, and we mostly see their undersides. However, I am almost entirely certain they are in the genus of Springtail. There's a few species that look really similar and are found around here, but which one of them we have in our ecosystem, I really don't know. I found a really big one of those structures, which pretty much completely rules out the possibility of these being springtail poops. What on earth could these be then? Maybe some weird algae? These are all clumped around an algae slime blob. Time for an aesthetic beauty break starring baby bloodleaf leaves. In the past, we saw how the population of the rotifers and the springtails both appeared seemingly out of nowhere, and growing rapidly due to a lack of competition and predators, and an abundance of food. This usually results in boom and bust cycles, where populations grow so quickly they deplete resources, like food, light or space, forming their own competition and ultimately crashing. After a while, resources are present again and the few survivors can cause a rapid population growth again. A similar pattern can be seen with predator-prey cycles. And indeed, in this jar, we saw the rotifer population boom and then bust. We also saw the springtail population boom, but never bust. So, what's up with that? Well, that is an excellent question and I wish I had a similarly excellent answer. Luckily for those of you who love an unresolved mystery, I don't. I think there's simply much more food available to the springtails compared to food availability for the rotifers. After all, the terrestrial part of this closed ecosystem is much bigger than the aquatic part. I never even intended for there to be an aquatic part, but these jars have a will of their own and this one decided that it wanted to be a paludarium instead of a terrarium, which is just fine with me. And to be fair, I did add a lot of moisture in this jar in the form of the banana peel, the smoothie and the water in the plant pot. I even added a bit more water to the ecosystem a few days after it was made, because the plants were wilting. Looking back, I don't think the plants wilting was due to drought, but something else, potentially even too much water. Luckily, I got away with it because both plants are still miraculously alive today. Anyway, those are the sources of all the water on the bottom of the jar today. Wait, what was I talking about? Something about animals or whatever? Oh right, I was talking about how the springtails probably had more resources. Sorry guys, this happens sometimes, but you've probably noticed that by now. Oh, right. While it may be so that there were or are more resources for the springtails, you'd expect their population to grow as large as those increased resources allow, and then bust. But that also didn't happen for some reason. Maybe some other factor is capping the population size of the springtails, preventing them from becoming their own competition. Maybe something like a predator. Maybe something like a predatory mite. Oh yeah baby, perfect segue, woohoo! But predators need to be alive in order to predate, and for whatever reason, I've only been able to find dead mites. And also a minor detail, I've never actually seen a mite prey on a springtail in here, so I don't actually know if these are predatory mites in the first place, which is great for keeping things vague and unresolvable. So why are all these mites dead then? Another excellent question. Based on the fact that they don't seem to have decomposed at all, I'd say that they died relatively recently. They also all seem to be engulfed in water droplets, so it's quite possible that the environment is too humid for them, or that they're even literally drowning in the water on the glass, because they're not well adapted to wet surfaces, contrary to the springtails. I feel another excellent question coming. Shouldn't the amount of water in this ecosystem be constant because it's completely closed airtight? Why yes, of course. The amount of water within the jar remains constant, but the distribution of water within the jar might not. Because very interestingly, this jar appears to have some sort of water cycle. Over the years, I've noticed that sometimes it seems to be quite dry, while other times it's very humid. Right now it's very wet, with a lot of water on the glass. Part of this has to do with temperature, as hotter air can hold more water. At the time of filming it's October and getting colder. Another part of the equation is plant growth. Plants are almost entirely made from water. If there's more plant material, there's less water free to wet the jar. As we've seen, the amount of plant material fluctuates quite a bit. Right now the bloodleaf has a lot of little leaves, while the coffee plant is not that large at all. The same is also true, by the way, for the amount of animal life, in the form of springtails and mites in this case, but I think that effect is negligible. Pretty interesting, right? And here I finally found a living mite again, a very small one, probably just born. So if it is indeed the moisture that's trapping some of them, well, it luckily isn't trapping all of them. As this little mite kindly directed us towards the water, let's see what's living in there. 3 years ago, the rotifer population had indeed busted, but they weren't extinct yet. Now, I'm sorry to say, it looks like they are. I've looked around for quite a bit and haven't managed to see a single one. I don't recall ever seeing their population boom again in the past 3 years, so maybe the ones we saw were the last survivors of that population bust, which perished not much later. Sadly, I don't quite remember the last time I actually saw a rotifer. It could have been about 3 years ago, or maybe a small population held on for a little bit longer. I just don't recall. That certainly doesn't mean there isn't any life in the water though. Just look at the color. It's absolutely filled with algae. This moat around the plant pot also acts like something analogous to a plant, just like the slime on the sides of the jar. All these algae also must have grown from spores in the soil in the plant pot. Even though I never intended for it to be present, this layer of water filled with algae makes me very happy. Well, that is the 4-year-old fruit fly jar. Initially envisioned as a closed terrarium, it actually turned into a closed paludarium over time, albeit with a small aquatic part. Perhaps it is partially because of this paludariumization that this closed ecosystem has thrived for so long by now. In my experience, closed terrestrial ecosystems that remain closed airtight permanently have a very hard time staying alive, much more so than aquatic ecosystems. So far, I've only had one closed terrarium that managed to survive for years, being the big native closed terrarium, and one part terrarium, the big paludarium ecosystem. So it's really special to see a closed ecosystem that at least started out as a terrarium do so well. Of course, it's always fascinating to see life closed off in a relatively very small space, completely separated from the larger ecosystem we call the Earth, survive and thrive for years. This project of course started out as a closed ecosystem with fruit flies. Those lived and reproduced in this jar for several months, ultimately going extinct. Maybe I'll try this again in the future, but add way less fruit fly food to see if they'll fare well for longer. Even though the fruit flies did go extinct, the ecosystem itself kept developing, with quite a lot of new organisms appearing and forming large populations, although that development has logically slowed down over time. Even after 4 years, this closed ecosystem is very biologically active. For years now, there's been a springtail population in this jar that seems to be ecologically stable, either because of possible predation by mites or through some other mechanism. So, ecologically stable population dynamics are possible, even on such a small scale. All in all, this has proven to be a fascinating project, which of course I will keep updating you about. So, if you haven't already, well, you're going to have to subscribe. For now, dear viewing tube children, it is time to say goodbye to this glass encapsulated miracle of life. And I will put it back on the windowsill with all the other projects, but this time facing the other direction to see what impact that'll have. Goodbye.